students, today we are going to know about cell biology unit 1. Definition: a cell is the smallest basic unit. We all know that the cell is the only basic unit of the, it is responsible for all the life process, and it is the structural and functional unit. And first it was discovered by the Robert Hooke. He is the first biologist. And the study in the study of cell has been made only possible with the light microscope. At first in 1665, the Robert Hooke, with the help of light microscope, he discovered the cell by through a piece of cork. We all know this is the cork. When he get a section of this cork, he observed this type of cells. With the help of the light microscope, he observed this type of small cavities and surrounded by a foam walls, and he used this term as the cell. And later Leeuwenhoek, he also observed nucleus in erythrocytes through with the improved microscope. Differences in the prokaryotes, eukaryotes, a type of cell, it's unicellular, and eukaryotes are unicellular and multicellular. The cell size ranges from 0.2 to 2.0 μm in diameter. In eukaryotes, it is 10 μm to 100 μm. And cell wall usually present, and it is chemically complex in nature. In eukaryotes, if it's present, it is chemically simple in nature. In prokaryotes, nucleus is absent. In eukaryotes, it is present. Ribosomes are present and smaller and spherical in size, and in eukaryotes they are present and larger in size and linear in shape. The DNA arrangement in prokaryotes are circular, and in eukaryotes they are linear. Mitochondria is absent in prokaryotes and present in eukaryotes. Cytoplasm present but cell organelles absent in prokaryotes. In eukaryotes it is present and cell organelles also present. Endoplasmic reticulum is absent and present. Plasmids. In plasmid we can observe plasmids, ribosomes, lysosomes, cell division. How the cell division is occur in prokaryotes through binded fission and through mitosis. Biphasal, these are smaller in size, these are larger in size. In asexual, and these are both asexual and sexual. These examples are bacteria and archaea. These are plant and animal cell. Virus, these are the tiny smaller, they are tiny, which is smaller in size, like about 30 to 50 nanometers. These microbes are belongs to the family Viridae and the genus Virus, and they could not be placed in any kingdom because they are cannot practically neither living or nor dead. So these cannot be placed in any kingdom. So the term virus is coined by the Dutch microbiologist Martinus William Bujanic in 1897. He derived from the Latin, which means the virus means poison or venomous substance. These are the non-cellular, and it is enclosed around a protective envelope. The presence of spike, here we can observe. These are the spike around the body. This is the protective envelope. It is covered by a protective envelope, and these are the spike which help to attach to any host of the cell. It also carries a viral genome. We can observe here the viral genome is present inside the virus. This is the protective envelope, and this is the viral genome is also covered with a nucleocapsid membrane. And this is the virus, this is the nuclear envelope, it is enclosed in a nuclear envelope, and these are the spikes which are help to attach to the host. Next, electronic microscopic structure of animal cell. This is the animal cell, which is a type of eukaryotic cell which lacks cell wall and has a two membrane bound nucleus along with the other cell organelles. The animal cells will range from smaller few microscopic microns to the few millimeters. Like the largest animal cell we can observe in the complete earth means it is ostrich egg, which have this stretch over 5.1 inches across and weighs about 1.4 kilograms. The shape of the animal cell will be vary from flat, oval, rod shaped, and there are also different shapes like curved, spherical, concave and rectangular, and all these type of cells only we can observe under a light microscope only. Also that the animal cells of the eukaryotes, these also contain membrane bound nucleus, and other cells also present, and DNA also present in this type of animal cells, and they also likewise they contain membrane bound organelles present. They have DNA and membrane bound nucleus, and each membrane bound organelle have their function. They carry a specific function. This is the structure of the animal cell. This is the nucleus, and this is the endoplasmic reticulum. This yellow part is called as endoplasmic reticulum. The blue, all over the blue part, is called cytoplasm. On the cytoplasm we can observe the cell organelles. These are the cell membrane bound organelles. Next, this is the fourth one. This is called as the Golgi apparatus. This one is the mitochondria. This one is the ribosome. Seventh one is the lysosome, and this is the wire. In nucleus, inside the nucleus we can observe the genes like DNA, and next the outer protective layer is called the cell membrane. Next, the animal cells have this type of parts like cell membrane, nucleus, nuclear membrane, centrosome, lysosome. Cell membrane is a membrane layer which consists of lipids and proteins, and its function is to control the entry and exit of nutrients inside the body. It controls the entry and exit of the nutrients and other microscopic entities. And next, nucleus, it is organelles that contain several it has the suborganelles like nucleolus, nucleosomes and chromatins. It also contains DNA and other genetic materials. Nuclear membrane, it is a double membrane structure that surrounds, it is present around the nucleus, and it also referred to as nuclear envelope. Centrosome, which is present near the nucleus, and it have the microtubules. And lysosome, these are the round organelles which surrounded by a membrane. These lysosomes consist of a digestive enzymes which help in digestion, excretion and cell renewal process. Cytoplasm, in picture we have observed the blue color jelly-like material. It is the jelly-like material. In the jelly-like material, all the cell organelles are present. And next, Golgi apparatus, it is a flat, smooth layer sac-like organelle which is present near the nucleus and involved in manufacturing, storing, packing and transporting the particles throughout the cell. Mitochondria, we all know that it is the powerhouse of the cell, which means it releases the energy. Ribosome, these ribosomes are made up of RNA-rich cytoplasmic granules which help in protein synthesis. And next, endoplasmic reticulum, these are thin, and we can observe near the nucleus a winding network, folded network will be present, that is called as endoplasmic reticulum. Next, vacuole, a vacuole is a membrane bound organelle present inside the cell. It helps in the maintaining shape and storing water, food, waste, etc. And nucleopore, these are the tiny holes present in the nuclear membrane. The nuclear membrane is present around the nucleus, and these are the tiny holes present on this membrane is called as nucleopore, which help in the movement of nucleic acid proteins across the membrane. The structure of plasma membrane. A plasma membrane mainly consists of four types: carbohydrates, phospholipids, proteins and conjugated molecules. They are about of the length of 5 to 8 nanometers. The plasma membrane is flexible and lipid bilayer, which means the lipid layers are present in 2 layers. So this is called as the lipid bilayer. So and it contains the cytoplasm of the cell. By this structure, they have described as the fluid mosaic virus, because the arrangement of the molecules and by this 2 layer structure of this plasma membrane, it is also described as the fluid mosaic virus, and it was first proposed by the American biologist Garth L. Nicholson and Seymour Jonathan Singer in 1972. He also describes that the plasma membrane contains of these components: phospholipids, proteins, carbohydrates and cholesterol, and these all collectively gives a appearance like fluid. So that's why it is described as fluid mosaic virus. It is a phospholipid layer, like we have earlier know that it has the layers of two phospholipid, phosphate group and lipid groups are present, two layers, and these lipids have with the phosphate group. The phospholipids have one head and two tails. Here we can observe this fluid. This is the plasma membrane. Here this is the phospholipids. This has one head and each one has two legs. These are the glycoproteins. These are the integral protein. This is the protein channel. This helps to movement of certain particles from the outside to the inside, or outside to the inside, or inside to the outside. These are the hydrophilic tails, and this is the cytoplasm. Next, functions of plasma membrane. The first function is as a physical barrier. It controls the exit and entry of the other particles. That's why it helps as a physical barrier of the cell. The selective permeability, which means only some components like water, oxygen, carbon dioxide, these type of components can easily travel through the plasma membrane, but some of the polar contents like sodium, potassium, these ions cannot pass through this plasma membrane. So these can pass through only through a proper channel called a protein channel. So by these specific channels, these type of sodium and potassium can pass through from one side to the other side. Endocytosis and exocytosis. If a cell ingests a larger contents into the cell body, they create like a cell into the body, and it was a cell taken into large quantities and the whole bacteria from the extracellular fluid. When a cell releases, which means endo means it intake of the outside material. Exocytosis means releasing of a material. This is called as endocytosis and exocytosis. And next, cell signaling. In cell signaling, we can observe that the signaling means facilitate communication. It gives communication and gives signaling between the cells. Next, structure of Golgi complex. This is the structure of Golgi complex, which is present in the cell cytoplasm. It can we can observe both in plant and animals. It has these are the cisternae. These are called as cisternae. We can observe that about 5 to 8 cisternae are present, which is a cup-like. Here we can observe these are the cup-like compartments. These are flat and disc-shaped and 20 stacks per cell. The functions is the protein packaging. It provides with proteins. It has membrane-bound vesicles which help to transport from one place to the other, including lysosomes, plasma membrane and secretion. They also play a role in lipid transport and formation of lysosomes. And in plant cells, this Golgi apparatus synthesizes complex polysaccharides that make up the cell wall. Next, structure of endoplasmic reticulum. It is one of the important cells in the cell. Whereas the nucleus function is to serve as a cell membrane, ER acts as a manufacturing and packaging system. ER membrane anatomically divides into cisternae and sheaths. Cisternae are structurally tubular, and in mammals they are 50 nm in diameter, and in yeast they are 30 nm in diameter. This is the endoplasmic reticulum. We can observe two types, smooth and rough endoplasmic reticulum. Why this is called as rough endoplasmic reticulum? The ribosomes are attached to this membrane, outer membrane of the endoplasmic reticulum. That's why it is called as the rough endoplasmic reticulum. It is helpful for the transport of carbohydrates, proteins, and and it also provide a wide area for the cellular reactions. And its main important role is to synthesize lipids, glycogen, proteins, and also steroids like progesterone, testosterone, and cholesterol. Next, structure and function of lysosomes. These are the tiny sacs filled with fluid containing enzymes. These are the cells which contains enzymes which enable the cell to process its nutrients and are also responsible for destroying the cell after its died. It break down digestion in materials inside the cell, break down digestion of materials outside the cell. It is called inside the cell, which means called as autophagy, and from the digestion outside the cell it is called as heterophyte. And next, structure and function of ribosomes. Ribosomes is the most important cell organelle composed of RNA, like 70S ribosomes and 80S ribosomes we can observe. These are free and bound ribosomes which are similar and associated with protein synthesis. Ribosomes are helpful for mainly protein synthesis. Next, mitochondria. We all know that it is double membrane, outer membrane. Here we can observe. This is the outer membrane, there is the inner membrane. This is called the matrix. These are the powerhouse of the cell as it releases energy to the cell. It helps in detoxifying ammonia in the liver cells and helps to promote the growth of new cells, and also regulates the metabolic activity. And it is the responsible for building certain parts of the blood and various hormones like testosterone and estrogen. And it also help in maintaining adequate concentration of calcium ions and compartment of cell. It is also involved in cell signaling, cell senescence, controlling the cell cycle. Next, structure and function of nucleus. It is the present inside the cell organelles. The nucleus is completely bound by membranes. It is encased by a structure referred to as nuclear envelope. The membrane distinguishes the cytoplasm from the contents of the nucleus. In the nucleus, we can observe the main concept. DNA is present inside the chromosome. Chromosomes are present inside the cell. These are the chromosome, chromatin material, which means the genetic material called like chromosomes. In chromosome, we can observe the DNA, which is the genetic information which can transfer from one parent to the other. The nucleus controls and regulates the activities of cell like growth and metabolism, and carries genes, structures that contain the hereditary information. Nuclei are the small bodies that are often seen within the nucleus. Thank you.